Well, hello, everyone. Welcome back to Neuromatch 3.0. Very, very pleased to have this keynote session this morning featuring Sean Escola from Columbia University in theme E on technology and tools. Sean did all of his training at Columbia University, including an MD, PhD there, and he's now an assistant professor at Columbia of psychiatry, I believe. Is that right, Sean?
Yeah, that's right.
Excellent. So very excited to hear him talk to us today about thalamic control of cortical dynamics for sequence behaviors. Sean, go ahead.
Okay, thanks a lot, Chris. It's really great to be here, everyone. I'm looking forward to telling you about some work that I've been doing for the last couple of years with postdoc Laurel and Logiaku, who I'll have a slide on her in a second. So the idea is that we're interested in how thalamus, which itself is controlled by inhibitory input from the basal ganglia, can in turn control the dynamics in cortex, which, as I'll show, will be important for the generation of sequence behaviors. Let's see here. Okay, so this is Laurel. We have the paper that I'm going to be presenting all the results from out on the bioRxiv. It's in review, actually in revision, and so hopefully it'll be out soon. And I wrote a tweet print maybe about 6 months ago or so. So if anybody wants to, you can probably dig it up. So imagine you've got a scenario where you're interested in generating some behavior, and say this time trace might be the activity of a muscle over time, and it could be related to, say, some task of reaching over and moving, typing on your keyboard or whatever it is. And at some other time, you want to produce some other behavior, so some other pattern of activity on the same muscle. What you'll notice is that actually this is a sequence. And so this is a hypothesis that we're using as a starting point, assumption actually, we're using as a starting point, which is that complex continuous behavior can actually be decomposed into small stereotyped behavioral primitives, which themselves then can be sequenced together to give rise to long complex patterns of behavior. So if you think about production of behavior as production of sequences, then it raises the question of the necessary underlying computations to get this done. So here's a list of our behavioral elements, which we'll call motifs. And we want to put them into some sequence, as I showed you before. And so here are the computations that we need to be able to solve, or the way I think about it anyway, which I think is useful. First, there's the question of selection. So which motifs will you generate and in which order? Then there's the question of transition. How do I get from the pink motif to the olive motif in a meaningful way and don't have some weird big blip in my behavioral output? And then there's the fact that each of these motifs is still a dynamical thing that is executed in time. So how do we execute these things once we've selected and then transitioned to them? So I'm going to focus on transition and execution. In fact, I'll speak mostly about execution and then a little bit about transition later. We're kicking the can a bit on selection, at least in the context of this talk. Okay, so here you have a set of sequences. And now let's imagine that we've got a new motif that we want to learn and add to our library. But one thing that's really important, so this is sort of a constraint on our problem, is non-interference. So we need to make sure that whatever motif, when we learn a new motif, we don't risk overriding motifs that we already know. And then we want to be able to take that new motif and stick it sort of arbitrarily in a new sequence. So we need to be able to transition from this olive to this orange motif and from this orange to this purple motif on the kind of the first time that we've ever done it without ever having learned those specific transitions. And that's related to this idea of flexibility. So the ability to compose these motifs in arbitrary orders. So these are two constraints that we want to put on our model. So here's everything I've said so far. So we need to be able to do selection, transition, and execution. And we want to be constrained by non-interference and the ability to be flexible. Okay, so this is the motor system. And of course, in the schematic of the human brain. And it's an extremely complicated system with many different interacting areas. We're going to focus on the motor cortex, the basal ganglia, and the thalamus. in the context of this presentation. People have studied the loop between motor cortex, basal ganglia, and thalamus, and back to motor cortex as a major loop involved in action selection. In fact, our model will be consistent with that, but we will focus also on the fact that motor cortex is reciprocally connected to thalamus. So it sends feedback to motor thalamus, and as well as gets input from motor thalamus. And so this, so using the anatomy, these anatomical constraints, we'll say that for our model, that cortex and thalamus are reciprocally connected. Additionally, basal ganglia inputs into the thalamus and inhibits it. The output cells from the basal ganglia are inhibitory cells. And then finally, of course, cortex is recurrent. And importantly, it will turn out, especially for this non-interference constraint, the thalamus itself is non-recurrent in terms of excitatory connectivity. Okay, so this is our a simple schematic then. We've got this recurrent cortex sending projections to some output neuron. It itself is bidirectionally connected to thalamus via these thalamocortical and cortico-thalamic connections. And the thalamic cells receive input from the basal ganglia, inhibitory input from the basal ganglia. Okay, so we can also turn to the physiology. By the way, I think it would be great to have people jump in if anybody wants to. I'll try to keep monitoring the chat, but it might even be easier if you put something in the Q&A, because then it will definitely flash up on my screen. I'd be happy to answer questions as we go along. So we can also look to the physiology as a way to get additional constraints on the model. In Mark Church's lab and Christian Chenoy's lab, there's been, for a long time, people have been studying reaching tasks in monkeys, and many others as well in other labs. And we can think maybe of one of these reaches as a motif in the context of our framework here of thinking about sequence production. In a seminal study in Nature several years ago, Mark had monkeys make many different reaches. So these are all different reach conditions here with these different colors. So this is sort of the XY axis of where the monkey's reaching. And when they analyze large numbers of neurons in the motor cortex and then subject it to some linear dimensionality reduction techniques, they see a very interesting result, which is that just before the reach commences in this low-dimensional space, depending on the reach condition, you're in a different spot of this low-dimensional space. But once the reach initiates, you follow these rotational dynamics, which is simple linear dynamics. And so we're going to actually take this a step farther and assume, although there's other data to support this, that within each of our motifs, and maybe this is even the definition of what becomes a behavioral motif in our context, within each of our motifs, the brain is acting linearly. And that the nonlinearity is at these transitions between motifs. And furthermore, just as Mark noted is extremely important for these reaching behaviors, preparation in our system will be a major dictate of what the ultimate output of the behavior. So we can go a little further and we can look at some other studies. So in the Costa lab, they've studied for several years in several studies, including in this one, 2014, having rodents press levers. When we look at this paper, I'm extracting away some of the details of the data now. They've identified output cells from the basal ganglia that during this sequence of presses, and a single one of these sequences is what we would call a motif in the context of this paper. During this motif of lever pressing, you see some of these output cells of the basal ganglia are inhibited the entire time of the motif, and others are sustained and active throughout the motif. And actually, it's a substantial percentage of the cells that are in these output nuclei of the basal ganglia that are in either these inhibited or these sustained states. Let's see, there's a question here. A new motif does not overwrite older ones. What about driving a manual versus automatic car where the overlearned sequence manual here can perturb the new sequences in an automatic car? Well, I mean, I think it's a good question. There's definitely some possibility for overwriting, but I don't know. I don't know about that exact result. I don't know if that's a common thing that you have difficulty once you've gone from one to the other. But certainly the example that we use, and maybe it's worth mentioning briefly here, is dance. So in professional dancers, in ballet or whatever, you can learn a series of dance moves, and you can use them in different dances, Swan Lake or some some other Romeo and Juliet. And really the difference between those dances is just which order you do some of the same moves in. And you could learn maybe as you get more and more advanced in dance some new dance move, and you can start to incorporate it into the dances that you've been learning to produce without overriding the ones you already have. So this is this idea that we have of increasing your library without overriding. We'll get to our mechanistic ideas about this in a second, and you can tell me whether or not you think it's a good idea. Okay, so here in the Costa data, we say that we have the possibility for motif-specific input. So during the course of a motif, this pattern of inhibition coming from the basal ganglia can be specific to the current motif. And then lastly, the basal ganglia can silence thalamus. So the input from thalamus can be strong. And so what we mean by that, and there's many studies that have looked at this, and so what we mean by that is that if a particular input from the basal ganglia to one of these thalamic cells is active, then that cell kind of drops out of the cortex thalamus system entirely. So that's the major nonlinearity in our system. Okay, so how do we solve the non-interference problem? Well, the way we'll do it, it's actually seems quite simple, but ultimately is a prediction of our model that we can discuss. We think that the basal ganglia inputs are non-overlapping. So from motif to motif. So if you're going to produce, say, the pink motif, and you want this pattern of activity, you want to release the neurons that are driving the pink dynamics while inhibiting all the others. So that drops everybody else out of the dynamics except for the pink guys. Whereas if you're doing the olive behavior, you want to do the opposite. You want to drop everyone else out of the activity except the olive guys. So you don't want to inhibit them in order for this set of dynamics to be active. And then finally, if you want to guarantee non-interference, you can only do learning in these cortico-thalamic and thalamocortical synapses. And so that's a final assumption that we'll make, and it means that really cortex is acting as a reservoir in our setting. Okay, so here's the simple dynamics within a single motif. It's a simple linear dynamics. We've got some network time constant tau, and then we've got a set of weights. We've got the intracortical weights, this JCC. We've got these thalamocortical weights, this JCT, and we've got these corticothalamic weights, this JCT. But we've got this funny term here, S mu, which I will describe now. It's a selection matrix that implements our basal ganglia inhibition. So imagine you want to do pattern 2, mu equals 2 here, and you want to inhibit everybody except for this olive neuron. Well, then you can just put zeros on the diagonal except for at position 2, and that will zero out all of these inputs coming from cortex to thalamus except for the one that passes through the olive neuron. So that means that this, so this is, like I said, this is the key nonlinearity in our model. And that means that this J tilde mu is an effective connectivity for pattern for motif mu. So for every motif, you have a different effective connectivity in cortex. And this term, this JCT S mu JTC, is a low rank perturbation to the cortical connectivity that's different for every motif mu. Okay, so let's talk about our motifs for a minute. So the interesting thing about transient motifs is that you can write them as sums of exponentially modulated sines and cosines. Well, that's true even for very, very, very long motifs. But in the case of transients, you can do it for with a very small number of components, even if t is sort of relatively long. So say 100 times the size of cortex. In this case, we have these patterns here, and they're 100 times our cortical timescale, and we can sum them all up with these exponentially modulated sines and cosines, only 6 of them per pattern. So what's the interesting thing about this? Let's try to get some insight. I want to do a little bit of a digression here to talk about what does it mean for a network to be able to generate an output. So I showed you that these outputs could be generated by a sum of a small number of these wiggles. And so what does a network need to do to generate an output? Well, it needs the right dynamics. And what does that mean? So let me do a brief detour. And what I mean by the right dynamics is that the network dynamics are these wiggles that are available to the network to sum up and produce outputs. So the dynamics are these exponentially modulated sines and cosines. There's a technical term for them, which is the eigenmodes. In a linear network, the wiggles are fixed. In a nonlinear network, they can actually change over time, depending on the current activity of the network. And it's the eigenvalues of this effective connectivity matrix, J tilde mu, that define the wiggle. So here's a particular set of eigenvalues. Eigenvalues just tell you the frequency that each wiggle oscillates at on the y-axis and how quickly it decays on the x-axis. In this case, we're always dealing with stable networks. Okay, so here's this is actually the same network, and now I've color-coded, I've highlighted a few of these eigenmodes. And so let's look at just the population of all of the wiggles this network can produce. And so here's just a subset of them. I didn't put them all over here, but you can see I've got some of the pink and olive ones in here. And if these are our target patterns, well, it turns out that from this network, if we sum up these wiggles with the correct coefficients, and the coefficients, this is an important point, are set by the initial conditions. This gets back to Mark's data that the initial conditions were very important for the preparation, execution of motor behaviors. So the coefficients on these wiggles, these eigenmodes of your network, are determined by your initial conditions. And there's a one-to-one relationship. And if you sum them up, then sure enough, you can get what you want. And the coefficients can be small. And this is important because if there's a relationship between, there's this relationship I said between the coefficients and the activity in the network. And if you have really large coefficients, that means you have really unreasonable, a large neural activity in order to produce your network. And that's the next example I'm going to show you. So let's say we want to make this sort of triangle wave pattern now from the same network. Well, we can try to find what are the coefficients that will allow us to add up all of these guys in order to produce the output. And we can do pretty good. But in order to do that, we need coefficients that are order a million times larger than the ones we needed for this pink and this olive one. So that's not very good. And if we constrain our coefficients to be small, then we do a much, much worse job. But of course, there's some other network that has some other set of eigenvalues, and therefore some other set of wiggles, these eigenmodes. And if you sum up just a small number of those, as I've shown you, then you can get the right pattern with small coefficients, which means small activity in the network. So in order to generate the right dynamics, it's important to note that having a large network is not enough. So I could have, these have 200 nodes using 200 eigenvalues. I could have taken this to infinity, and I still would have had these really large coefficients that were necessary in order to sum up these relatively large coefficients in order to create these patterns well, because the correct dynamics are not present in the network. So let's come back to our problem. What we want to do is we want to take our cortico-basal ganglia thalamic system and see if we can get the right dynamics into the system. So let's look at the most constrained version of this problem. where we have just a single neuron in thalamus that's released by the basal ganglia and can interact with cortex. And in this setting where we have just this single thalamic loop, so this is just a rank 1 perturbation to the cortical network, what Lurlin was able to show, and it's pretty straightforward if you do the calculation, is that there's an equation that that relates U, the thalamocortical weights, and V, the corticothalamic weights, such that if you have a set of desired eigenvalues that you know, if you add up the eigenmodes associated with those eigenvalues, you'll get the output you want, then you can be sure that those eigenvalues will be present in your network. In fact, so here, this is the equation. We have this term P. This will come up in a second, where we take the pseudo inverse of it. And P is just defined as having these terms that are related by the desired eigenvalue, the eigenvalues that are related to those wiggles that you want to have in your network, and the eigenvalues that are related to the wiggles that you sort of have before you add any thalamic interaction. And this L term up here, just the eigenvectors, the left eigenvectors of the cortical network. Okay, so we can do that, right? So we can use this equation, and if we do that for these 4 different patterns, we can see that what I've shown you here in pink and then olive, purple, and orange are the desired eigenvalues that we want. It's a little hard to see, but if you squint, you might be able to see. In black are the eigenvalues that the network has before any thalamic interaction is added. And they're the same. The black is the same for all 4 of these. And then after we use this equation and relate the corticothalamic weights according to this equation, then we do in fact see that every single one of these different circles has a blue eigenvalue in it. The blue eigenvalues are the eigenvalues of the effective cortical connectivity J tilde mu. And after making these perturbations and setting the initial conditions correctly, then we get the right output. Okay. So what are the constraints to make sure that this works? So we'll go through a couple of them quickly. If you recall from this equation, I said that the pseudo inverse of P is an important point, and this P has to do with is related to these desired and these original eigenvalues. And so what we can see is we can do some numerical experiments to sort of understand what's going on here. And so what I've done is I've taken a network of size 500, and I've increased the number of desired eigenvalues from 32 to 50. And I've tried to use my equation, and I see that when it's 32, I do just fine. I'm able to get the desired eigenvalues that I wanted. But when it's 50, I fail. And that's due to numerical precision, a failure in my computer. It's not due to the fact that the equation is wrong, but the fact that the numerical precision is bad is a property of P. And these terms start to become correlated. You start to have correlated rows in P that's related to how many eigenvalues that you're desired eigenvalues that you're trying to put into the network. Similarly, if instead your desired eigenvalues are far away from the initial set that you started with your cortical network, then you can do okay if you have a small number. But if you increase now to 24, which is much less than actually how I did before with 32, then again, I have a failure. And finally, we can, of course, reduce the size of the cortical network. So this changes, again, the ratio between the number of desired eigenvalues and the size of the cortical network. And I do okay if for a smaller network if k is 24, but if I increase it to 34, then they fail. The reason I chose these specific numbers of k was to try to make the condition value of p constant across these roughly constant across these rows. So what's the take home here is if there are few desired eigenvalues and those desired eigenvalues are close to JCC, then this will work. And the good news is, as I said before, that for transient motifs, even ones that don't look anything like combinations of signs, you can do a good job with just a small number of wiggles, small number of eigenlines. Okay, so the next thing that we wanted to look at was noise robustness. And here's the deal. First, just focus on these dotted lines. When I have no noise, of course, I do great. I'm able to produce my output. But if I add just 5% noise, maybe this is even 1%, I forget, in this particular plot, it doesn't really matter. I have a massive amount of noise that's added to my, I do nothing like what I want. But it turns out that in my equation, I only used half of the parameters. I set V given some random U. But if I optimize U for noise robustness, then once I've optimized, even after adding noise, I have perfect performance. And the difference between these bottom two is just the scale. I can look at another one of my patterns. This one's much, much worse. Before I optimize, when I use a random U, I have terrible sensitivity to noise. But as soon as I do this optimization, then I can have a very, very limited, it's essentially no perturbation with respect to noise. And so what's going on here? Well, it turns out that during this optimization process, so maybe I should start by saying the dynamics in this network are highly non-normal once you add this rank 1 perturbation. You have a, and what Laurelin was able to show is that the direction of noise amplification is aligned both with the readout and with the directions of the wiggles that you want to use to compose the readout. So that means if you have small perturbations, then you have massive amounts of noise along these directions. And this is sort of the base case that you would expect when you use a random U. But as soon as you train U, what you're doing is you're finding a geometry in the space of the network dynamics such that the direction of noise amplification is quasi or orthogonalized with the readout and the directions of the wiggles that are needed to compose the readout. So this is an interesting idea that weights are needed. One way that weights can be used to provide noise robustness is by orthogonalizing directions of noise amplification from the directions that you care about. Okay, so next we wanted to look at how we do these transitions, right? So everything I've shown you so far is how we execute individual motifs, but how do we get from motif A to motif B, from say the pink motif to the olive motif? And what Laurelin did is she recognized that there was a possibility of adding another population of neurons in this corticothalamic loop. And this population of neurons, we would see if we could use this population to speed the dynamics by which this cortical activity would converge to some baseline. And so since the dynamics are stable, always stable, we know that we're going to get eventually to 0. But if we add these preparatory this preparatory network, this cortical thalamic preparatory network, can we get the dynamics to go to 0, 0 much, much faster than otherwise would? And that's what I'm showing here. So it turns out that we are able to do that. We can optimize the corticothalamic weights of these preparatory cells, and we can increase the time of convergence from something that might take many times the network time constant to something that takes just about less than 1 time the network time constant. The network time constant here it takes about 5 to 10 times the network time constant, whereas before it might take 100 times or more. And the way this works is that this preparatory network that we add to the dynamics changes the eigenvalues in the cortical network such that they are far from the stability line. And so this speeds the relaxation to 0. And now, since the dynamics are well, okay, let me stop there. I'll come back to another point in a second. And so how does this work across different sizes of this thalamic preparation network? So how many thalamic neurons do you need to speed up your convergence of your cortical dynamics? And so what I'm showing with these different lines here is the size of the thalamic population as a ratio of the size of the cortical population. So in this plot on the upper left, the blue line is 3 neurons because the cortical population is 100 neurons, up to 50 neurons for the pink line, and so forth. But let's focus on the upper left first. And what this is showing is that if I have a, say, if we look at the green line, which is the one that we'll ultimately use, so this is a thalamic population of 10% of the cortical population. If I have a thalamic preparatory network that's 10% of the cortical population, then I can get my cortical activity down to 5% or 1% of its initial value within 5 to 10 neural time constants. And the data is exactly the same for networks of larger size. And in the bottom right, I'm just showing you all of the networks now on a log scale. And so you can see that it's really the ratio of the thalamic size to the cortical size that dictates how quickly you can speed up your dynamics with one of these preparatory networks. So what we'll do is we'll use this preparatory network that speeds the dynamics to with 10% of the thalamic cells in terms of the size of the cortical network, such that we can decay down to 0 as quickly as possible. But instead of decaying to 0, I'm going to go to the next slide really quick. Instead of decaying to 0, we can decay to the starting value of the next motif if we add an input to the cortical dynamics. So this is now from the paper. I'm sorry for the change of format. So if we inhibit all of the motif-specific thalamic cells, and we release this preparatory circuit, and we give an input to the cortical network, then we can very quickly decay to the exact necessary preparatory state for the upcoming motif. And so over here, what I'm showing you on panel B is the readout, which starts at some random value and quickly decays to something near its starting point for the upcoming motif. And also the cortical activities in the final row that start at random different values, but very quickly decay to their initial state to generate the dynamics for the upcoming motif. And then we switch, the basal ganglia switches, it turns off the preparatory network, it turns on the network, the thalamic unit associated with the upcoming motif and generates the upcoming motif. And so if you put this all together, you can then very quickly and seamlessly transition between motifs. And if we look at the ones that I had shown you before, sorry, the slides are not quite polished. I was still working on these this morning. If we look at the ones I showed you before, these brief preparatory periods in gray are when this preparatory activity, this preparatory network is on, and you have these quick transitions that allow you to be set up and ready to generate the next dynamics. So essentially, the way to think about this whole system that we're proposing is that the basal ganglia is essentially playing the keyboard that is the thalamus. The basal ganglia is the maestro, and it's on the keyboard, and it's inhibiting different thalamic cells in order to affect the dynamics in cortex, in order to cause different output. And in between each of these different motifs of behavior, one of the chords, if you will, that the basal ganglia plays on the thalamus is this chord that releases this preparatory state. that has these which then causes these very, very fast dynamics of cortex to quickly prepare for the upcoming movement. And then it goes on and plays the chord that's associated with the next motif. So that's what I wanted to show you guys this morning. I want to quickly mention a couple of the predictions that are associated with this model. So importantly, if you perturb the basal ganglia or the cortical inputs to thalamus, you would have differential effects. So if you change the input to thalamus from the basal ganglia, that should affect which motif you're going to be performing. Whereas if you perturb the cortical input to thalamus, that should affect motif execution dynamics. Additionally, we're proposing that there are motif-specific subpopulations. The simplest version of this is that the motif-specific subpopulations are in thalamus itself, and that's kind of how we built the model. But it's also possible that they may be in cortex, and that there's a effective feed-forward step where you go from thalamus to cortex to an isolated population that then projects more broadly in cortex. And then perhaps the most interesting prediction that we're making is that motor preparation is always needed. In order to solve this transition problem, you always need to go through this preparatory process, even when you're doing fast movements. And this is in contrast to the standard view that if you're going to be performing some sequence of behavior, you should learn to chunk that behavior and produce a single chunk of activity very quickly. But instead, we're proposing that if that sequence is composed of elements that you would otherwise use, then the way to perform that sequence is by having motor preparation, quick motor preparation between each sequence element. And I just want to end by noting that that's what's been seen in some very recent new data, very exciting data by Andrew Zimnick in the Churchland lab. So what they did is they trained a monkey to do a sequence of reaches. And there were two conditions that the monkey did this sequence. Either it did them as fast as possible, so A and then B, or it was told to have a delay between the two. So do A and then wait and then do B. The hypothesis, well, I don't want to say what their hypothesis was because I'm not sure, but an hypothesis that you may have had is that when you had that delay and you got to sit at the end of A for a while before moving on to B, that you might get to prepare, and that's what they show here. So when you go so what I'm showing you here is the activity that's in the preparatory dimensions in cortex. And what you see is, ignore the stars. I forgot to remove these. And what you see is that there's a lot of activity in the preparatory dimension during the activity just before the second reach, when you have this delayed condition, which makes sense. So you have that you're going to wait for a while before you reach again, and so you have a lot of activity in the preparatory dimension. That's for both monkeys. Whereas in the immediate condition, you might have assumed that you wouldn't have a lot of activity in the preparatory dimensions prior to the second reach, but you do. Even though you're moving as quickly as possible, A and then B, you see that you prepare for the first reach, that's here, and then you prepare for the second reach in the middle of your sequence. And so that's what I wanted to show you is this relatively recent data. Okay, once again, Laurelyn Logiaco is the one who did the primary work. And sorry for the roughness of some of those final slides. And I'm really excited to take any of your questions.
All right. So sorry, we're in a virtual format and you have to deal with the solo clap from me instead of the audience, but that was a great talk, Sean. Lots of discussion and questions popping up. We've got a few different types of questions in just a few minutes. So what I think I might do is try and combine together a couple of themes. I think one theme that kind of popped up in some of the discussion and in the Q&A box is about the connections between what you're talking about and reservoir computing. You talked about it's not enough to just have a large network. You have to have the right motifs in it. And I feel a little confused about the relationship between that and these reservoir sort of ideas, where with a large, richly connected network, you could have almost any possible theme or motif. The question was about basal ganglia, and I know you said a different role for basal ganglia, so it could also apply to cortex here. What do you think about the role of kind of reservoir sort of ideas for these motifs in either basal ganglia or cortex?
Yeah, so I was intentionally provocative with that middle section, and anticipating this question. So so when you say train a reservoir network in order, like you using say a force training or something like this in order to produce some output, the recurrence that's required as part of that training process gives you an effective change in the connectivity. So it's similar to this idea of the loop between cortex and thalamus that we're talking about. But there's no way in that system to have these different motifs that can coexist unless you hard code them all in with the same loop, right? So the feedback loop that you have in the standard reservoir setting will guarantee that you're going to have interference between motifs. Whereas in our case, we have sort of an isolated feedback loop. I mean, I think this is I really like this project, and I think that there's a lot of really interesting, cool things about it. But there's some things that are like sort of also very sort of almost simple and obvious, right? So if you have a difference, if you are able to isolate the components of your network, then you can be sure that you'll have non-interference. So that's one comment I want to make here. That's related to that idea.
Great. Well, great talk. Thank you, Forzan, for that talk. Another talk looks like Conrad chimed in with a question about how identifiable these models are from physiological data. Any comments on that?
Yeah. So certainly this is an incredible simplification of the true complexity of the motor system. But we do think that some of the predictions that the model makes are testable and should be looked at. So one of the things that we're most interested in is looking at what happens when you are able to isolate the corticothalamic projections and perturb them in the setting of some behavior. So our hypothesis is that this idea of the basal ganglia playing the keyboard that is the thalamus, and by doing so setting up the dynamic reservoir for the production behavior, is only possible because of this feedback between cortex and thalamus. And so I think that if we were able to look at the consequences of interacting with that particular connection in this network, and people have looked the other way, right? So people have looked at the consequences. So Adam Hantman, for example, has recently looked at the interactions between thalamus and cortex and shown that they're very, very important in the context of different behaviors. But that's sort of maybe a little bit less surprising given the dogma.
Yeah, great. Well, I think maybe time for just one more short one. Barbara Fulner had a number of questions in the Q&A that were great, but one really resonated with something that I was going to ask myself, which is, in your thinking here, your motifs are discrete and you need to transition between them. And I think if you could just talk a little bit about the decision to model things that way, rather than thinking about more graduated or continuous combinations of motifs being the kind of primitive to model with. Can you explain a little bit about your decision making there for that modeling?
Yeah, so there's a couple of different points. One is that you actually, and I tried to get at this in my little my digression in the middle. If you have the right dynamics set up in your network, then you can produce a number of different motifs with the same dynamics. And in fact, that's what Mark would say is going on in motor cortex during reaching. Right, you can actually fit the same linear dynamical system to a handful of different reaches. And so that suggests that one dynamics is sufficient as long as you're able to set up the initial conditions correctly. So that would suggest that actually the entire, so it's kind of funny because under that hypothesis, the only thing that really matters is this preparation itself.
Yeah.
And so what I'm saying, what you were sort of calling out as potentially a funny part of, do we need to have this dedicated transition module? Well, that transition module is the thing that would also allow you to do a range of different behaviors in the setting where you're sharing the dynamics, because it's all about setting up initial conditions.
Yeah.
Now, whether or not if you had a set of, say, two motifs that really have very different underlying compositions, what wiggles you need to build them out of, could you have some kind of gradual change in the dynamics by slowly changing which thalamocortical loops you bring online or take offline, or scale them in some way? I'm sure there's some possibility there, but it's not something that we've had the opportunity to look at in detail.
Okay. Great. Well, sadly, we are out of time for the discussion, but Sean, the talk was fantastic. Thank you, everyone watching, for chiming in with your questions and in the chat. Sean, really appreciate your time. Thanks again very much.
Yeah, and I saw Laurelyn in the chat. Sounds like she was answering a lot of questions. So I meant to mention she's on the job market soon. So keep your ear open if there's an opportunity there. All right, thanks everyone.
Fantastic. All right, thank you.